this lesson, we're going to look at how substances move across plasma membranes. So we call that the transport of substances across membranes. And in this session, we're going to look at diffusion and something called facilitated diffusion. Now, before we go any further, we need to have a little think about some background information. One important point that we need to think about are solutions. So solutions are mixtures of two substances, solutes and solvents, and usually just one solvent. Solutes are substances that are dissolved in a solvent. So an example could be sugar, and an example is that sugar is dissolved in water. And in a solution, which of course is a liquid, the solute molecules and the solvent molecules have kinetic energy. They are constantly moving around. And this is very, very important. So as these substances are moving around, they are bouncing off each other and colliding with each other, and slowly they would move apart. This is diffusion. As the solute molecules move in a solution, they bump into each other and the solvent molecules. Over time, they will get further apart. They will spread out. So if we have one molecule here in a high concentration, and over here that molecule is in a lower concentration, all these molecules are moving around, as are all these molecules, randomly. But because there's more of these here, then over time, the chances of those molecules moving from there to there is greater than the chances of the molecules here moving from there to there. Some of these molecules will move that way, but because there's more here, then they will move out and start to move to an area where there's less of them. So we have here a high concentration, and here we've got a low concentration. And we call this diffusion. And because we have a high concentration here initially and a low concentration there, then we call this a concentration or diffusion gradient. Now, the difference in these two concentrations is really important, because if there's a large difference in concentration, therefore the concentration gradient is very steep, then there's more molecules over here, and that means over time there's a greater chance of these molecules moving across here. If these two concentrations are very similar, this is only slightly higher than there, then the diffusion gradient is not as steep. It's not as great. And that means diffusion will take place at a slower rate. It'll still happen, but at a much, much slower rate. Now, the confusing thing is when we come to cells, because when we have a cell, a cell has got around it a plasma membrane. And that plasma membrane, if we have some molecules in here and some out here, that plasma membrane may allow some substances to pass through, but may not allow other substances to pass through. So these might be able to diffuse out. And these molecules, because they're in a higher concentration outside than inside, they can diffuse in. And diffusion takes place until both sides, either here and here or there and there, have the same concentration. So when both sides have the same concentration, then diffusion will stop. So diffusion is the net movement of molecules from a high concentration to a low concentration. Now, there's a number of factors that can affect the rate of diffusion. And those factors are, first of all, the surface area of the diffusion layer. So basically, if there is a large surface area, the rate of diffusion will be higher. So we can say the rate of diffusion is proportional to the surface area. Proportional means that as this increases, that increases the same. So if we double the surface area, we will double the rate of diffusion. The second feature is the concentration gradient, which is what we've got here. So the bigger the difference in the concentrations from where the molecules are moving to where they're going to is called the concentration gradient. So in this case, the rate of diffusion is proportional to the concentration gradient. The last factor that affects the rate of diffusion is the length of the diffusion pathway, or the thickness of the layer that the molecules have to travel. It's the distance that the molecules have to travel. So if we had these molecules here and we timed how long it would take them to get to here, and then we timed how long it would take them to get to right over here, of course, it would be faster, it would take less time for them to get to here than there. So therefore, the rate of diffusion isn't proportional to how far they have to travel, because as that increases, the rate of diffusion will go down. So we don't say it's proportional to, we can say that the rate of diffusion is proportional to 1 over the length of the diffusion pathway. And so we say this is inversely proportional. So the rate of diffusion is inversely proportional to the length of the diffusion pathway. There are other factors that will affect how quickly substances can move across the membrane. So for example, we talked previously about some proteins that form little channels or carriers that allow substances to pass through the membranes. So if a membrane's got a lot of these in them, then that will also increase the rate of diffusion. But we're really interested in these 3 factors. And these 3 factors can be combined together into something called Fick's law. And Fick's law is this, that the rate of diffusion is proportional to the surface area where diffusion is taking place. It's also proportional to the concentration gradient, so we can put times the concentration gradient all over the length of the diffusion pathway. Well, we said here that rate of diffusion is inversely proportional to the length of the diffusion pathway, so 1 over, so we put that on here. So we put the length of the diffusion pathway. Now that is sometimes described as the thickness of the exchange surface. That's Fick's law. Now what is important here is that the rate of diffusion is not equal to that. So if you calculate the surface area, you calculate the concentration gradient, you measure the length of the diffusion pathway, and you put all those numbers into that, you wouldn't get a number that is the rate of diffusion. However, if you got a number and then you change one of these, if you doubled the surface area, then the number that you get then would be double the original one. So proportional to is not the same as equals to. So previously we've talked about diffusion, and diffusion is a net movement of molecules from a high concentration to a low concentration. However, if there is a membrane, as there are around the outside of cells, so the cell surface membrane, or membranes that make up mitochondria, endoplasmic reticulum, then these form barriers to some of these solutes, some of these substances that need to move across the membrane. Now that might be a problem for cells. Cells want to control what is going in and out of them, but at the same time they want to make sure that some substances are actually moving across the membrane. So what membranes have in them, as we previously discussed, are proteins that can act as channels or carriers. And there's lots of different types. And these help diffusion to take place across the membrane. And so this is called facilitated diffusion. The word facilitated means helping. So these use proteins in plasma membranes that help molecules to diffuse. And so There are a number of different types, but one type we'll talk about are carrier proteins. Now, carrier proteins are proteins that span across the membrane, and they react to the presence of a specific molecule. So if we have a protein, it might look a little bit like this. So there's our protein, and I'm not going to draw all of the plasma membrane because it will take quite a long time to do, but there's our phospholipid bilayer there, and this here is a carrier protein, and this is going to allow a molecule to pass across. Here is our molecule. When that molecule enters that little area there, it binds to that protein in the membrane. That then makes that protein change shape. Now the energy for that comes from the kinetic energy of that molecule. So that molecule goes into there, it makes that protein change shape and opens up the other side to that molecule, and then the molecule can move out. It's sometimes called the ping pong effect, where the molecule goes in, ping, and then pong comes out. Now what is important here, there's a couple of things that are important. No extra energy is provided to the molecules. The only energy required for this to happen is the kinetic energy that this molecule has. As it moves into here, it basically makes this change shape and allows that to go through. So that's the first thing that is really important. The other thing that is really important is that only this molecule can enter and make this protein change shape so it can pass through. So it means that these protein carriers are specific to the molecule. It doesn't allow any old molecule to get through. It has to be a particular molecule that will enter that site and allow it to pass through. They are carrier channels. We've also got protein channels. So we've got carrier proteins, and then we've got protein channels. Protein channels also are intrinsic proteins, so they span the membrane, and they usually have a channel which is hydrophilic. So if you imagine that's a protein that's spanning that membrane, there's our phospholipid bilayer. It has got a channel, and that channel is usually hydrophilic. Remember, the middle of membranes are hydrophobic. They've got those fatty acid tails there, and that's going to stop water but other hydrophilic substances passing through. And in a solution, the majority of solutes are going to be in cells hydrophilic because they can dissolve in water. So these substances are not going to find it easy to diffuse across the membrane. However, here is a little channel which is hydrophilic. And that channel is hydrophilic because, if you remember, proteins are made of amino acids. And different amino acids have different R groups. Some of those R groups are hydrophilic. So all the amino acids that are found along the edge of that channel all have hydrophilic R groups. So it makes this area hydrophilic. So basically, if we have a molecule and it's in high concentration over here and low concentration here, then it will want to move across by diffusion, and it can't over here because there's a layer of hydrophobic fatty acid tails. But here there is a channel which is hydrophilic, and so the substances can move across down their concentration gradient and across the membrane. Now once again, these channels don't let any random molecule pass through. Most are designed to allow only the passage of particular specific molecules to go across. And this means, once again, that cells and cell membranes have the ability to select what moves across.